Good morning or good afternoon. Today I'd like to discuss some aspects of the neuromuscular junction. It will be a relatively short screencast, and I will present some of the essentials of how the communication between the motor nerve and the muscle, the myocyte, takes place. Now, you've seen this schematic before, and we discussed several of the processes that take place. For instance, if tissue is damaged and you may experience pain. Today we will mainly focus on this element, so this aspect, which is the communication between the peripheral motor nerve and the muscle. And there's a special dedicated structure between the nerve and the muscle, which is called the neuromuscular junction. And clearly it is essential that the process functions properly in order to move around, to talk. In essence, it's one of the key components also of a proper output. And we discussed previously that our motor system is the most important output system of our body. Now, you've seen this schematic previously as well, but it's always good to repeat some of those aspects. We discussed information transfer from the primary motor cortex as descending volleys of action potentials in the pyramidal tract to the second motor neuron, the lower motor neuron, the alpha motor neuron in the anterior horn of the spinal cord, where it synapses, and this lower motor neuron transmits signals to the muscle. And here we have this dedicated connection, which is known as the neuromuscular junction or motor end plate zone. And actually, this is an example of a peripheral chemical synapse. We discussed synapses previously as well. There are many of those in the central nervous system, but clearly we have synapses as well in the peripheral nervous system. And it's an essential structural component of intercellular communication. And just as a short recap, it's good to note for completeness that in addition to chemical synapses, where the space between the two structures, a neuron or another neuron or a neuron and a muscle, is typically of the order of 30 to 50 nanometers. And we know that there is a transmission delay of approximately 1, sometimes 0.5 millisecond because of the release of the neurotransmitter and the interaction with the postsynaptic receptors. We also have electrical synapses, gap junctions. They're mainly located in the central nervous system. The gaps are very small, of the order of 3 nanometers, and the transmission delay is essentially 0 milliseconds. You could view it as a passive resistor, essentially. It's not completely true because there are dynamical changes in gap junctions as well, but we will not discuss those. At the same time, because the gap junctions can be viewed as a resistor, the communication is essentially bidirectional. So you could view it as a reciprocal synapse, while most chemical synapses can be viewed as rectifiers. There are some exceptions to the rule where there's a bidirectional chemical synapse, but that is relatively uncommon. Most chemical synapses are unidirectional, and therefore the information transfer is essentially from A to B and not from B to A. If we zoom in to the neuromuscular junction, we find this beautiful structure. This is the presynaptic part, so this is the terminal axon, and here we find the vesicles where the acetylcholine, which is the neurotransmitter here, is stored. This is the membrane. You can find a vesicle here that is fused with the membrane. This is the gap, and this is the postsynaptic density, which is the myocyte, part of the myocyte. Now that vesicles are involved in this process, and that there is essentially quantal release of acetylcholine, because every time when a vesicle fuses with the axon, with the membrane, there's a release of a particular number of acetylcholine molecules. So in that sense, it is quantized, because every vesicle contains a particular number of acetylcholine molecules. And that was actually discovered in 1972 by Katz and Milady, who did beautiful experiments in this particular structure, and they were able to record basically relatively short duration changes in the membrane potential of the myocyte with characteristic shapes, and they deduced from that the release of acetylcholine should be quantized, so contained in most likely in vesicles. So this is a recording in rest, and this is a recording a little bit later where there is a deflection in the membrane potential of the receiving myocyte with a characteristic shape. It's almost a very fast increase in the membrane potential and then an exponential decay. And this results from the activation of a particular number of postsynaptic acetylcholine receptors that are mainly permeable when there is an interaction with the acetylcholine for sodium. There's some potassium that goes to the outside as well, but there's mainly sodium influx. So it's an excitatory phenomenon that we see here, and a reduction of the membrane potential. And here a zoom in of this picture I just showed. This was essentially measured in rest, because now and then it appears to be the case that acetylcholine molecules do fuse with the membrane, even if there's no action potential. So now and then there is a quantal release of acetylcholine, but only a small, relatively small number of molecules that induces a postsynaptic change in the membrane potential, but not sufficiently large that subsequently voltage-gated channels are being activated. So this is the essence. During rest, now and then there is a quantum, say a vesicle with acetylcholine molecules that is released, containing between 5 to 10,000 molecules. They bind to the acetylcholine receptors, and that induces a local end plate potential, which is also known as a miniature end plate potential, or an MEPP. So a small change in the postsynaptic membrane potential, but not sufficiently large to subsequently activate voltage-gated channels that are very close to the acetylcholine receptor to generate a muscle action potential. So during rest, that typically does not occur. And actually you can measure that with a needle in a muscle. So you've seen this picture before, a motor neuron axon and the connection with the chemical synapse to several muscle fibers. So this is the motor unit, a motor unit. And if you do a recording with a needle and you have some patients, then you can actually measure those miniature end plate potentials. And we did that many years ago. So this is time in milliseconds, and this is the voltage in microvolts. Please note that this is extracellular recording, so it's not as large the deflection as you would observe when you would measure in the myocyte. But you do see this quite characteristic changes in the extracellular potential, essentially reflecting miniature end plate potentials of the receiving myocyte. And so what essentially happens, and shown in the schematic here, this is rest. There's a release of one vesicle of acetylcholine molecules, like 5 to 10,000. There's interaction with a particular number of receptors, say 100. And slowly the acetylcholine is removed because it diffuses out. And there's acetylcholine esterase, an enzyme that breaks down the acetylcholine. So it is essentially removed from the cleft, and then gradually more and more channels close, so they don't stay open. So initially 100, for instance, then 70, and then 20, and this goes according to an exponential decay, approximately. Now, what happens if you stimulate a nerve, either electrically or voluntary, when you want to make a particular movement? Then, of course, there's an action potential drain, there's presynaptic opening of the voltage-gated calcium channels. Then there is release of many quanta of acetylcholine, say 60 vesicles. So there's relatively a lot of acetylcholine released in the cleft, and that will induce several miniature end plate potentials, and they do summate, temporal summation mainly, and that is typically sufficient to activate and open the voltage-gated ion channels, the voltage-gated sodium ion channels. And then an end plate potential is created, resulting in a motor action potential. So there will be an action potential traveling across the myocyte, and that will, on its turn, result in muscle contraction. Now, shown again in this schematic, presynaptic vesicle with acetylcholine released by calcium, and then there's an interaction with the receptor of the acetylcholine, and sodium will mainly go into the muscle. A little bit of potassium will flow out, but the most important effect is the influx of sodium. So it's a highly selective receptor, but not completely, and that will, if sufficiently large, induce eventually a muscle action potential. We can also show this in a little movie. Let me turn on the sound, and then I will show you the movie starting now.
At the neuromuscular junction, the presynaptic nerve ending contains membranous vesicles poised to release their neurotransmitter cargo, acetylcholine. Neuronal stimulation and membrane depolarization trigger a localized entry of calcium ions at specialized transmitter release sites called active zones. This calcium influx, in turn, initiates a chain of signaling events that drives the fusion of the vesicle membrane with the plasma lemma, a process facilitated by several neuronal proteins including SNAP25, VAMP, and syntaxin, which together constitute the snare complex. The tightly regulated membrane fusion results in the secretion of acetylcholine into the synaptic cleft. The acetylcholine molecules diffuse across the synaptic junction and bind directly to specific receptors on the muscle, leading to muscle contraction.
Okay. Now, there are several diseases associated with this neuromuscular junction, and the two most well known are myasthenia gravis and Lambert-Eaton myasthenic syndrome. So what happens in myasthenia gravis, those patients in most cases have suffer from an autoimmunity disease where antibodies are created that interact with the acetylcholine receptor, making the receptor in essence dysfunctional. So it results in fewer functional acetylcholine receptors, and that means that there is a lower safety factor for neuromuscular transmission. What do we mean with the safety factor? Well, typically there's a relatively large concentration increase of acetylcholine in the synaptic cleft, much larger than is needed to occupy all the acetylcholine receptors. However, if there are only a few receptors left, then the number of receptors might be insufficient to induce a sufficiently large membrane depolarization, and action potentials will not be generated. Myasthenia gravis is a postsynaptic disorder resulting from blockage of acetylcholine receptors. Another condition that is presynaptic is Lambert-Eaton syndrome, and in Lambert-Eaton there are, which is also an autoimmune disorder, there are antibodies directed against the voltage-gated calcium channels, which are on the presynaptic side. And that results in insufficient opening, or an insufficient number of calcium channels that opens in response to traveling action potentials. So the intraneuronal calcium concentration will not increase sufficiently large, and that results in a reduction of acetylcholine release. Also a reduction of the safety factor of neuromuscular transmission. Now finally, there are several drugs that we can use to treat a problem at the neuromuscular junction resulting from a relatively insufficient amount of acetylcholine, be it presynaptic release that is insufficient, as in Lambert-Eaton myasthenic syndrome, or the postsynaptic reduction of receptors. And we use this quite often in the clinic in patients with myasthenia gravis, and we have particular drugs that interfere with the acetylcholinesterase. So that is the drug that breaks down the acetylcholine in the cleft. So we have anti-cholinesterase agents, and they prolong excitation at the neuromuscular junction. So these drugs are pretty well known in the clinic to treat patients with myasthenia gravis, amongst others. For completeness, some insecticides also contain these anti-cholinesterase agents, because a lot of the bugs also depend on acetylcholine for proper functioning. And if you apply a relatively large concentration of the anti-cholinesterase, then this will result in an abnormal increase of the acetylcholine, and that interferes with muscle relaxation. So if the concentrations are very large, you will not be able to relax the muscle anymore, and the muscle will essentially remain in a cramping state, killing the bugs. And this is also being used in some nerve gases, for instance sarin. So in conclusion, we very shortly discussed this beautiful structure. This is again an electron microscopy image of the neuromuscular junction. This is presynaptic, a lot of the vesicles with the acetylcholine. This is the axon, this is the muscle, and this is the synaptic cleft, and this is the membrane where we find a lot of the acetylcholine receptors and voltage-gated channels. So in sum, and as a cartoon, this is what we shortly discussed. Nerve terminal with the voltage-gated calcium channels, shown here. Of course, the voltage-gated sodium and potassium channels that are all around the presynaptic nerve and in neurons in general to propagate the action potentials. The vesicles with the acetylcholine that, if released, release acetylcholine that can interact on the postsynaptic side with the acetylcholine receptor. And close to the acetylcholine receptors, we find another voltage-gated sodium channel, but now in the muscle. So if these receptors are activated sufficiently, the membrane potential will sufficiently depolarize that the voltage-gated sodium channel will become activated, and that will generate an action potential, a muscle action potential. Another protein that is indicated here is MuSK, muscle-specific kinase. This is also a particular protein that can be affected by autoimmune disorders, resulting in dysfunction of the postsynaptic part of the neuromuscular junction. We will not discuss it, but there are some patients with myasthenia gravis that do not have a problem with the acetylcholine receptor, but they have, or they create antibodies against the MuSK. In closing, we discussed neuromuscular junction, we discussed miniature end plate potentials and the relation with the muscle action potential, and very shortly addressed neurological disorders like myasthenia gravis and Lambert-Eaton myasthenic syndrome. More details are in the book, and we touched upon some treatments. Thank you.